Good afternoon. My name is Dr. John Thomas. I'm an associate professor of medicine within the Department of Genetics at the University of Texas, Rio Grande Valley. I will be conducting session 2 for the short course for the Center for Genomic Research. And today's topic will be virology and DNA viruses. So at the end of this session, and again, the way that we've designed these short course sessions are for people with a level of understanding that's between K through 12 educators and undergraduate students. Our purpose for building the short course sessions are ideally that the sessions will build on top of previous sessions. So session 1, we looked at a general overview of the immune system. Here in session 2, we'll do a basic examination of virology, which will then build on sessions 3 and 4, which will look at the genetics of virology in terms of how the immune system responds to viral infection. And then we'll look at some genetic diseases that are caused by misinterpretations of the immune system within the context of a viral infection. So for session 2 today, which is virology and DNA viruses, our basic learning objectives will be to kind of provide an understanding of what some of the general characteristics of a virus are, how viruses are categorized, and what type of infections we have within the context of virology. We'll discuss briefly the life cycle of a virus in terms of how the virus gets inside of a cell and begins to initiate infection. We'll discuss some of the basics of virus replication with a focus on DNA virus replication. And then we'll speak about and briefly examine some DNA viruses that are considered to be significant human pathogens. The image that we're seeing here is an image from the world's first electron microscope that was constructed in the early 1900s by Dmitry Inadovsky, who was a Russian virologist that really first started to kind of look at viruses and figure out what these things were. So let's start with the basic premise of the lecture itself. What is a virus? A virus is essentially a subcellular, I'm going to refer to it as an organism. There is a debate within virology as to whether or not viruses are living or dead, if you will, if they're alive or inert. But for the purpose of the discussion here, we're going to call a virus a subcellular organism that has a parasitic life cycle. It has no metabolic activity outside of the host cell and must rely on a host cell and the replicative machinery that's inside of the host cell to produce more viruses. And this is where we get the parasitic nature of the virus coming in. Viruses are the most numerous and ubiquitous forms of life on planet Earth. No other species of organism, if you will, on the planet outnumbers viruses. By our best estimates, there are somewhere more than 10 million trillion, this is 10 to the power of 31 viruses on this planet. Of course, this is impossible for us to verify, but we are discovering new species of viruses every year. Every place that we look, whether it's a virus that parasitizes mammals or insects or plants or bacteria, we see, or even other viruses, we seem to find viruses capable of infecting nearly every cell type that we've come across. So what makes a virus? When we say something is a virus or refer to something as a virus, what does this general definition include? At its most basic, a virus is essentially going to be a particle that we often refer to as a virion that's going to contain some kind of a nucleic acid genome inside. This will either be DNA or RNA. And this DNA or RNA molecule is generally going to be surrounded by some kind of a protein shell. And then this protein shell, which we refer to as the capsid, may itself be surrounded by secondary and tertiary membranes, which we'll talk about in a moment. But basically, it's going to be a piece of DNA or RNA inside of a physical structure that protects this molecule, this genomic molecule, and also allows it to move around inside of the host and preferentially be able to interact with uninfected cells and then infect the cell and produce new viruses. There are oftentimes a complex array of proteins that make up the virion that are involved in helping to stabilize interactions between the viral genomic molecule, whether it be DNA or RNA, and the capsid protein shell that oftentimes surrounds this, as well as stabilizing interactions between the capsid and the viral envelope itself. So there's many proteins that are involved in kind of contributing to the overall structure of the virus. And the way that some of these proteins are configured in a line helps give viruses some of their unique shapes, which we'll talk about here in just a moment. Viruses are going to have, as kind of indicating the cartoon here, kind of specific receptors on the outside that should be able to interact with receptors that are on the target cell. These are often going to recognize proteins or sugars, sometimes referred to as glycoproteins, glycosylated proteins, that may be contained on the host cell. You can also see some instances where viruses will pick up some of the membrane envelope from the host cell that are parasitized, and it may also contain some of these viral, host cell proteins that make up the membrane that the virus butted to. We'll talk more about this when we get to the replication cycle of a virus. The complexity of a virus is often going to be directly reflected by the size of the viral genome. We generally have RNA or DNA as our viral genome. It could be both, and it could be different variations of these molecules, which we'll talk about. But in general, we see RNA viruses have less complexity from a genomic perspective than DNA viruses. DNA viruses, not always, but on average, tend to bring in more genes. They have more genetic material, so they're going to encode for more proteins, and this is generally going to increase the complexity of the virus. The more information that the virus can encode, the more proteins it can make, and theoretically, this should give the virus more function within the host. So what are some types of viral infections? If we look at types of viral infections, we can generally break this down into 4 basic types. And we refer to these as lytic, a chronic or persistent infection, an infection that's transforming, or an infection that's latent. So when we look at these, what we generally associate with viruses is going to be the lytic replication cycle. This is where the virus enters the host, it gains access into a host cell, it replicates inside of that host cell, and eventually the viral replication process inside the host cell will lead to the death of the cell. The virus is then able to escape the cell either through a process called budding, which we'll talk about, or perhaps the cell goes through apoptosis, which is programmed cell death, or maybe goes through just a lytic phase where the cell just dies from an uncontrollable death cycle. But either way, this allows the virus to then generally escape the cell. Usually the lytic replication cycle is going to be very quick. For RNA viruses, you can usually see this process complete itself in about 2 to 4 hours. So this is generally going to be a very rapid killing of the cell and subsequent production of progeny virus. In a chronic or persistent infection, these are generally infections that we associate with somebody who has a long-term viral infection. So for example, hepatitis, Epstein-Barr virus, herpes virus, even HIV to some extent. These are all considered to be chronic long-term infections. There are other viruses as well that cause chronic long-term infections. I'm not naming all of them. But a persistent or chronic infection is generally going to be characterized by low levels of virus being produced that may or may not be detectable within the host, but the infection remains persistent. It's neither cleared by the immune system, nor is it resolved through the general production of the life cycle of the virus. In addition, we can also have viruses that replicate in a latent state. And again, this kind of has some crossover with what we talk about when we refer to chronic infections. A latent infection, again, this could be Epstein-Barr virus, this could be CMV, this could be varicella zoster virus, the virus that causes chickenpox in children and shingles in those that are 50 and over. These are viruses that have the ability to maintain the viral genome in an inactive state somewhere within a cell, within the host, and then through some mechanism that we largely still don't understand, these viruses are able to escape that latent phase, they become reactivated, and then they enter into a lytic cycle of replication where they start to replicate. This is, for example, something that we commonly see with herpes viruses. If we look at herpes simplex 1 virus and the physical effect of producing cold sores on the mouth, this doesn't happen all the time. There's usually some trigger, some environmental stress maybe, or some chemical stress that causes the virus to reactivate, and then herpes virus is infected in the neurons, it replicates in the neurons, and will actually start to produce a bit of the physical pathology that we see that we commonly associate with herpes virus infections, at least type 1 infections, which are going to be cold sores. And then for whatever reason, the virus replicates for a few weeks, it elicits its physical change, if you will, and then goes back into the latent phase. And then the last type of viral infection that we see is we refer to as a transforming life cycle. And these are going to be viruses when they get in and replicate within the cell, they're going to produce some kind of altered cell growth. And generally, when we see this, what's indicative of this is we find a part of the viral genome present. So, for example, this would be something like maybe Epstein-Barr virus, Kaposi sarcoma. These are viruses that can cause cancer, which is clinically defined as uncontrollable cell growth as a part of their replication cycle. So we have these 4 different types of viral infections: transforming, lytic, chronic or persistent infections, and then latent infections. And it's usually within the context of one of these 4 types of viral infection cycle that we're going to see the virus falling into. So when we look at classification of viruses in general, we have, and I said this a few minutes ago, and again, this is not 100%, but for the majority of viruses, they're going to fall in 1 or 2 categories. They either have a genome that's composed of DNA or their genome is composed of RNA. Now, these genomes could be a single strand of DNA or a single strand of RNA. They could be double-stranded. They could be, sometimes we see a complex of multiple segments of RNA in the case of influenza. Sometimes we even see viruses that have genomes that are part DNA and part RNA. So there's many configurations that the viral genome can take, but it's generally going to be composed of either DNA or RNA. If we go just a level above that in terms of looking at the overall structure of the virus itself, so this is going to be the physical structure that encapsulates or surrounds the genome, we generally look at these in terms of being referred to as either enveloped or non-enveloped. The term envelope refers to the outermost membrane that surrounds the virus, which is generally acquired through the process of viral egress. This is when the virus is ready to leave the infected cell. And oftentimes when this happens, for an enveloped virus, the virus will actually exit through the cell, through an area in the host cell that has a plasma membrane. This could not only be the plasma membrane at the outermost surface, this could be a membrane-bound organelle within the cell, such as the Golgi or one of the ERs. So the virus is going to bud through an organelle or through the plasma membrane, and when it leaves, it's going to actually pick up a piece of that membrane and take it with it. We refer to these viruses as enveloped viruses, and as shown in the graph on the, or as in the slide on the right, enveloped viruses, there's a considerable number of viral families within this enveloped category. We see the filoviruses. These are going to be the viruses that are such as Ebola virus, Marburg virus, the Rhabdoviridae. This is rabies virus. Paramyxoviridae, these are paramyxoviruses. The coronaviruses, we're all familiar with the recent pandemic caused by the SARS-CoV-2 coronavirus. It's an envelope virus. The orthomyxoviridae, influenza viruses, many viruses, herpes viruses, viruses that infect humans and mosquitoes, the flaviviruses, the alpha viruses. Many viruses contain this enveloped morphology and have this envelope membrane. We also have viruses that are non-enveloped. These are viruses that are not going to escape the cell from budding through a plasma membrane or a membrane-bound organelle, and they're generally going to be consisting of a protein shell that goes around the virus itself, that encapsulates the viral genome. If we look at the non-enveloped viruses, particularly viruses, again, that have some impact on human health, we're looking at things like polyomaviridase, papillomaviridase. This is the virus that causes genital warts. The reoviruses, adenoviruses, which infect a lot of people, may or may not cause infections. But we've generally got this breakdown of classification, not only in terms of what the genomic molecule consists of, but the outermost structure of the virus itself, whether or not it has an enveloped or it's a non-enveloped virus. And there's some benefits that come along with each one of these structures, which we'll talk about later here in the talk. If we look at viral genomes as a totality of what viruses we've identified so far fall into which box, and we look at DNA and RNA, by far the largest class of viruses, if you will, the largest genome classification are going to be single-stranded positive sense RNA viruses. When we refer to a genomic molecule being of a certain polarity, it's positive or it's negative sense, this is really referring to the organization of the genomic molecule itself. We'll talk more a little bit about why that matters, but for the purposes of our discussion here, a positive sense RNA viral genome is going to very closely mimic what we see in the host cell with regards to DNA replication. DNA makes RNA, makes protein. RNA transcripts are generally going to be organized in a 5' to 3' polarity, which is what positive sense RNA viruses are also organized as. And so this similarity to host cell RNA transcripts gives the virus some benefit in being able to hope that its RNA genome will be replicated. So if we look at this again from a numbers perspective, of the roughly 2,000 virus species that have been identified, the class 4s, the positive strand single sense RNA viruses, make up the largest family. And as it would happen to have, this is also the class where we see all of the mosquito transmitted, all the arthropod transmitted viruses fall within this category. So maybe it makes sense from a bit of an evolutionary perspective. Mosquitoes make up a very large segment of the insect population. Obviously, they take blood meals, so this gives them the ability to transfer the viral RNA if they're infected back and forth between humans and mosquitoes. If an uninfected mosquito feeds on an infected human, they could pick up that single-stranded RNA viral genome, amplify it inside of them, and then pass it on in their next blood meal. So from an evolutionary perspective, it makes sense that RNA viruses would make up a large family of viruses that we know about, since they leverage arthropods and the interaction between arthropods and mammals in the taking in consumption of the blood meal to pass along the genome. If we look at other classes, such as the negative strand single sense RNA viruses, this, for example, is rabies virus. Again, if you look at how rabies virus is transmitted, it really comes down to the process of mastication from an infected animal. So somebody or some animal gets infected with rabies virus, it's got to pass that along through a physical bite that's got enough virus inside of it to be able to pass it along. This is not going to be something that's going to probably be occurring as frequently in nature as an insect feeding on another organism and taking a blood meal. So the class 5 single-stranded negative sense RNA viruses aren't that large of a family. And as we go around the pie, we can kind of see this happens with other viral families as well. The viruses that are DNA, RNA, or RNA, DNA hybrids make up a very smaller population, followed by the second largest family, which is the double-stranded DNA viruses in class 1. And then we have single-stranded DNA viruses and double-stranded RNA viruses in classes 2 and 3. So overall, we've got a wide coverage of variations in RNA and viral genomes and what their makeup could be: double-stranded RNA, double-stranded DNA, single-stranded RNA or DNA. And of these 2,800 species that we're looking at, or 2,000 species that we're looking at, about 2 to 250 of these viruses are known to infect humans as either primary or secondary hosts. And again, when we look at that pie, a lot of those are going to be the class 4 single-stranded positive sense RNA viruses. So if we further try to stratify this based on how viruses replicate, we can see different strategies that viruses use based on their genome type to replicate, produce new viruses. And again, we've broken this down. This aligns with the classification system that was used in the previous slide, class 1, 2, 3, 4, 5, 6, 7. The same grouping applies over here. So for example, we look at class 4, the family that comprises the positive sense single-stranded RNA viruses. Again, these are the viruses that are going to infect through the bite of an arthropod, such as a mosquito, for example. Essentially what we see is that, and these are some representative types of this viral class 4 family, the flaviviruses. This is going to be dengue virus, Zika virus. These are yellow fever virus. These are flaviviruses. The coronaviruses we've already spoken about with SARS coronavirus. The coronavirus, these are going to be the poliovirus is a representative of the coronaviruses. The toga viruses are going to be things like the Western, Eastern, and Venezuelan equine encephalitis virus, Simbus virus, and leaky forest virus. These are all viruses that are transmitted through the bite of an infected mosquito. And as you would expect, we see, since the virus is coming in with a positive sense single-strand RNA genome, it's essentially a piece of infectious messenger RNA. Some, but not all, of the positive sense RNA viruses actually come with a genome that has a 5' cap and a poly A tail. As you may or may not remember from molecular biology, These are kind of critical components on a piece of positive sense RNA that are required to recruit ribosomes to that molecule of RNA so it can begin to produce proteins. Many of the positive sense RNA viruses either have a 5' cap and a 3' tail, or they have a structure that's very analogous to that, that's efficient at recruiting host cell ribosomes in the cell to the viral genome. Because remember, the virus cannot bring in these pieces to replicate its genome on its own. It has to rely on the machinery that's inside the host cell to be able to replicate its genome. So for the positive sense RNA viruses, for example, its genome is treated as basically as a piece of RNA. And so as we all remember from molecular biology, DNA makes RNA, makes protein. So in this case, we already have the RNA. The RNA is going to get transcribed straight into protein. Once the proteins are it's going to get translated into protein. Once this translation occurs, proteins are going to go through some modifications where they'll be assembled. And at the same time, the positive strand RNA genome is going to be used to make antisense copies, which will then be used to produce positive strand RNA copies. And both of these things, viral protein and viral genomes, will meet at these assembly sites where they'll be assembled into a virus and then be able to escape the cell and go on to affect other viruses. So you can kind of look through here and see some of the different strategies that are used. In the case of viruses that use a double-stranded DNA intermediate, for example, such as HIV, the retroviruses, they have a single-strand RNA genome that gets converted into a double-stranded RNA genome, which is then integrated into the host DNA. This integration eventually produces messenger RNA, which is used to make proteins and genomes, which will then go to assemble. So you can kind of look at some of the different strategies that the viruses all use, and for the most part, see that in a general sense, they're going to follow the classic dogma of DNA to RNA to protein that we see in molecular biology. Some of the viruses are able to shortcut that approach by already having an RNA genome, for example. So it maybe gives them a bit of an advantage from a time perspective in terms of being able to start to produce infectious virus quickly. One thing I think it's important to keep in mind as we think about viruses replicating inside of a cell is that this becomes a numbers game. At the same time the virus is inside the cell trying to replicate, the host immune system is trying to clear the viral infection. So on one hand, you have the virus trying to replicate to a sufficient density to be able to spread within the host. And on the other hand, you have the host immune system trying to come in and eliminate the infection. And so this oftentimes comes down to how fast can the virus replicate? How much virions can it be produced so it has a chance to spread its infection to another host before the immune system shuts down the infection. Even if you only get a few hours of advantage between an RNA and a DNA virus, if you're a class 4 virus coming in as a piece of messenger RNA already, for many intents and purposes, you're going to save time in being able to avoid the conversion from DNA to RNA because you're already RNA in that classic dogma of biology where we see DNA makes RNA makes protein. So there's probably a bit of a replication advantage that the RNA viruses have. And we do see this in vivo and in vitro. RNA viruses replicate to much higher levels, and they replicate to these higher levels much faster than DNA viruses do. So if we look at viral infection as a graph, we basically can break this down into 4 phases. We have the initial phase, which is referred to as the initial infection. The amount of virus is going to be very low inside the host. The virus has just gotten in. It's just started the infection. There's going to be a bit of a drop from the infection called the eclipse phase. This is where the virus is uncoated. It's beginning to make all of its viral proteins, but it's not yet got everything completed. This usually can last anywhere from a few hours to a few days. Again, for RNA viruses, single-strand positive sense RNA viruses that are vectored by arthropods, this process starts to happen in hours. And then once we exit this eclipse phase where we have enough viral proteins being produced inside of the cell, we then enter what's called the logarithmic phase, which is then the generation of new viruses, and the number of new viruses inside the host cell is going to begin to increase exponentially. So it's going to go up logarithmically. Eventually, the log phase is going to hit a plateau. You've started to theoretically exhaust all of the resources inside of the host cell. Perhaps the host cell is beginning to break down because all of the processes that the host cell would normally be engaged in to survive, making new host proteins, plugging holes in the membrane, shuttling resources around, all those processes are largely going to shut down because they're going to be taken over by the virus. So eventually, the host cell is going to run out of ways to keep itself going because all of its resources have been absconded by the virus. So eventually you're going to start to see this logarithmic phase begin to plateau. And when this happens, this is going to start to quickly lead to cell death. And in this phase, you would ideally see the virus leaving the cell to go on and infect other cells. If we look at viral infection, we can really break it down into three main phases: attachment, penetration, and uncoating. So in the first phase, attachment, as demonstrated by the cartoon here, we're essentially going to have a virus that's going to come in. It's going to recognize a host cell on the outside using receptors that the virus has to recognize and bind to host cell membrane receptors. Once this interaction phase happens, the virus is now attached. It will then move into the next phase of infection, which is called penetration. So the virus is attached. Generally, what we see is we see the formation of clathrin. This is going to be our clathrin-coated vesicle, where now the virus has been internalized with the receptors that it's bound to. It's been coated with clathrin, and now it's going to be fully internalized and brought inside of the cell. Following attachment, the next phase that we see is called penetration. This is actually going to be, not only has the virus now been attached and brought in to the cell, it's now inside the cell, but now we've got to start to see whether or not we can get the viral genome and the structure that might surround the viral genome broken down so we can start to get the viral genome released inside of the host cytosol so it can begin to be accessed by the host cell's DNA and RNA replicated machinery. So again, we have, in the case of an envelope virus in the top cartoon, the virus is recognized to the host cell receptor. It's now been attached. It's beginning to come in. Our clathrin-coated vesicle has now been broken down. And now we've got the viral genome being released. It's still covered with our capsid here, but the viral genome and the capsid structure have now been released into the cytosol. Sometimes we see the viral, when the virus comes in, it gets digested a little bit and broken down, and this will further weaken the capsid protein structure around the viral genome, again, getting it ready for replication. In the case of a non-envelope virus, we're going to have a similar process occur where the virus is going to bind, it's going to be internalized, and now we're going to have this breakdown occur. Usually there's going to be, perhaps in some viruses, we'll see a fusion event where there's a fusion between the viral envelope and a membrane envelope within the host cell, which will then allow for the release of the viral genome. Sometimes this process can be a process where we see acidification in a vacuole, or it can be an acidification independent process. But generally, we're going to have this penetration phase occur where now we've got the viral genome beginning to be exposed to the host cell replicated machinery. And the last phase, called uncoating, we now have our viral genome. It's been completely exposed. There's no more capsid protein. And now, if you picture out, this is actually not the outside of the cell. This is, we have our attachment penetration helping here. This is the outside of the cell. I'm sorry, right here is the inside of our cell. We've now got our genome being exposed. In this example, it's a piece of messenger RNA. It's now going to go through the host cell replicated process where we're going to see viral proteins being produced. Eventually, the production of viral proteins are going to lead to the production of more genomes. We now have our viruses being assembled and they're free to leave the cell and go on and infect new cells. So the viral infection cycle can basically be broken down into one of attachment, penetration, and then uncoating of the genome. These three phases really kind of, from a 10,000-foot view, describe what a viral infection is going to be like. So if we look at some of the cell structures that are involved in viral replication, and I've indicated some of these on the graph here or the slide, so I won't go through the names of all of them, but you can see the role that each one of these will play in helping the virus replicate. So for example, the cytoplasm. This is where inside the host cell, all the enzymes and basic building blocks for viral synthesis are going to be contained. We have the Golgi, which is going to be assisting the viral glycoproteins in their glycosylation so they can get properly formatted. So when they are used by the virus to infect new cells, they'll be functional. We have the nucleus where we're going to have not only DNA replication occurring, but for the RNA viruses that use caps or that need a cap for their genome, this is where they will get some of those caps. We have the endoplasmic reticulum. Of course, we all know about the rough and the smooth ER, where we're going to have protein folding, protein processing, transport. Some envelope viruses will bud through the ER and through the Golgi, so this could be a source of viral membranes. And then we have the plasma membrane, cell surface structures, and some other things here. So there's some general cell structures involved in viral replication. And this is why when we refer to viruses as being a parasitic organism, where this comes from. If these structures are not present, the virus can't replicate. So the virus has to be able to parasitize a host cell and go through the attachment, penetration, uncoating process to be able to gain access to these resources. If these cell structures are not there, virus replication doesn't occur. And we've actually done some targeted studies where folks have been able to go in and use cells that are deficient in a Golgi, for example, or they've been able to go in and block the activity of some of these organelles and infect them with viruses and look to see how viral replications occur. And in many of these examples, replication either doesn't occur or it occurs at such a low level that for all intents and purposes, viral replication really didn't occur. So these cell structures have got to be present for the virus to be able to produce new viruses and go on and continue the infection. So once all the virus proteins have been assembled, the virus has been given its new genome, it's got everything it needs to go, it's got to get out of the cell. If we're talking about viruses infecting eukaryotic cells, viruses that are infecting mammals, yeasts, insects, this is generally going to happen as a process of budding, where the virus will maybe bud through the cell if it's an envelope virus, and it will take some of the host cell membrane. If we're looking at viruses that infect bacteria, which are called bacteriophages, they do something that's called basically bursting the cell, which is shown in the images up here, where for all intents and purposes, the virus basically bursts a hole in the cell and is able to escape. It's a lot more complex than this, but for a 10,000-foot description of the process, the virus basically forces the cell to push the virus out, if you will. For eukaryotic cells, for viruses that are affecting mammals, insects, for example, it's going to be a little bit more complex. But essentially what you're going to see you're going to see all the viral proteins that are used to infect new cells are going to be structured and arrayed at a very specific part of the membrane where the virus plans to exit. So this is analogous to you putting your shoes at your front door before you walk out of the house. You want to make sure that before you leave, you can put your shoes on so you can leave. The virus is doing the same thing with its viral proteins. It organizes them at very specific areas on the membrane it's going to escape through, so it makes sure when it walks through that door, it can take those viral proteins with it. So once this process occurs, and this is commonly referred to as budding, the virus will bud through that area, through that membrane that contains the mature viral proteins. And once this budding process is complete, we now have a fully mature virus. We refer to them as being mature virions now that are fully capable to go on and infect other cells. So the virus has to attach to an uninfected cell. It's got to penetrate, get inside. It needs to uncoat the genome, replicate all of its proteins, get everything assembled at the area that it's going to escape from the cell, and actually egress/escape from the cell where it now is mature and it can go out there and infect other cells within the host. So taking a step back and just kind of looking at what some differences are between viruses from a very broad perspective, we can talk about differences between DNA viruses and RNA viruses. And I already mentioned some of this in some of the previous slides, but I think probably the biggest difference between DNA and RNA viruses are going to be how they replicate and what kind of factors come along when they do replicate and the speed at which these things can replicate. So I'd already kind of mentioned the speed, and that in general, RNA viruses seem to replicate a little bit faster than DNA viruses once the replication process has begun. And you could probably generally ascribe this to the fact that, again, DNA viruses have to first convert their genome into RNA, and then it can be converted into protein and then used to build new DNA viruses, where an RNA virus is already an RNA molecule as a genome. It can directly transition into producing viral proteins, and then making new viruses. So generally, we see RNA viruses replicate a little bit faster than DNA viruses. DNA viruses tend to be more complex because they're DNA viruses. They generally are going to have larger genomes. They generally are going to, as a result of that, bring in more genes. And these genes usually give the virus some advantage in being able to regulate the host cell or the immune system in the host. Some viruses in particular, such as the herpes viruses and the pox viruses such as smallpox, raccoon pox, canary pox, alligator pox. There's a pox virus that infects virtually every species of life, rabbit pox on this planet. But if we look at these large DNA viruses using herpes viruses and pox viruses, they bring in about 150 genes. Most of those genes are not used to produce new viruses directly. What they're used to do is to produce proteins that can regulate the host immune response, that can dampen or suppress the host immune response, which will allow the virus time to replicate. So DNA viruses are usually going to be a little bit more complex. They're going to bring in more DNA. This DNA usually gives them some benefit in being able to replicate. And the final difference that we see, and again, this is not 100%, but in general, DNA viruses, because they're a DNA molecule, and this is a dogma of molecular biology, must replicate in the nucleus of the cell. RNA viruses, because they're an RNA molecule, are going to replicate in the cytoplasm of the cell. Now, are there some exceptions to this rule? There are, and I mentioned them in here with the large DNA viruses using a second nucleus outside the original nucleus. But by and large, the big differences between DNA and RNA viruses are going to be the site of replication, the speed at which these viruses can replicate, and the complexity of their genome. A fourth difference, which is, kind of a here and there, is that RNA viruses are usually the only viruses that we see that are vectored by arthropods. So if you look at insects and the mosquitoes, for example, or biting flies, generally they are only going to be transmitting RNA viral pathogens through their bites. Is there an exception? There is at least one that's been described in the literature, and this is African swine fever virus, which is a DNA virus that is conferred through a biting insect, but it's a non-human pathogen. So there are always going to be exceptions out there, but in general, RNA viruses are vectored by biting insects, by arthropods. DNA viruses are not. So this just kind of is a brief list of some of the viruses that are of human significance. You can kind of go through the list here and look at some of these. They're broken down by genome type, double-stranded DNA, single-stranded DNA, segmented genomes. If it's RNA, is it one molecule of RNA? Is it two? If it's a double-stranded molecule. Over here are the members of the viral family, and then some information in terms of how big are they, what are some of the human diseases that they cause, and some of the primary hosts. And you can see for most of these host types, you're seeing mammals, humans, birds, insects, lots of insects. Some of these are fish viruses down here, some primates, and then some reptiles. If we look at the viruses themselves over here, herpes virus, papilloma virus, the virus that causes warts, cervical cancer. Viruses, polyoma viridae, causes progressive multifocal leukoencephalopathy, going to cause a form of leukemia. The pox viruses, smallpox. We look at other viruses down here. The positive sense RNA viruses, we're talking about things such as dengue virus, the flavivirus is right here, dengue virus, yellow fever, hepatitis viruses, polio virus, the viruses that cause rubella. So you can kind of look through here and see that although this list is not all-encompassing, there's probably some viral families on here with names that you recognize. The rhabdoviridae, the virus that causes rabies, measles, mumps, hemorrhagic fever viruses. This is going to be things like Ebola and Marburg virus that we hear so much about in the news coming out of these big outbreaks in Africa. So this is just kind of to give you an idea of what some of these viruses are of human medical significance. Again, this is not an all-encompassing list, but it's kind of something that lets you know that regardless of the genome type or even the organization of the genome, there's probably a virus out there that's capable of infecting humans and causing some kind of significant human disease. So these last few slides, we're just going to look at some basic examples of some of these things to kind of give you an idea for what some of these are. So Adenoviruses, these are viruses that infect a lot of the population. Many of us don't even know that we're infected with adenoviruses. In most people that are healthy and non-immunocompromised, you have a functioning immune system. You're not suffering from something like maybe lupus or rheumatoid arthritis or some other kind of severe autoimmune disease. You probably have an adenovirus infection and you may not even know it. Again, there are viruses out there that infect a large portion of the population. Adenovirus is one. Cytomegalovirus is a virus that affects about 95, 96% of the population. And many of us have these viruses and don't even know because they normally don't cause disease in a healthy, non-immunocompromised individual. So these slides are just here to kind of give you an idea of take a look at some of these things. The hepatitis viruses gives you some information on their genome, on whether or not there's a vaccine, there's any antiviral drugs present. The herpes viruses, which we hear so much about, herpes virus type 1 and type 2, very large complex double-stranded DNA viruses. Papillomavirus, we do have a vaccine for the warts, for cervical cancer and for cervical warts. The HPV 6-7 vaccine that's offered in many places, including here in Texas. Parvovirus, we do see some human parvovirus infections. We see a lot more parvovirus infections in dogs. Canine parvovirus is in particular a big problem for dog lovers. The polyomaviruses, these are going to be viruses that cause tumors, can cause and lead to cancer, including certain forms of leukemia. Pox viruses, we talked a little about smallpox. Again, there's a pox virus for virtually every species on the planet. I did a lot of work when I worked in industry with raccoon pox virus. There is a pox virus that's out there for salmon. There are pox viruses for monkey. We've all heard of monkeypox and the concern that monkeypox caused a few years ago, where we're seeing a lot of monkeypox transmission. Monkeypox can be transmitted sexually, so we had a lot of concern about monkeypox a few years ago. A lot of pox viruses out there. These are very large DNA viruses that bring an incredible amount of genes with them. You can see the genome size ranges anywhere from 130,000 kilobases to 375,000 kilobases. These are big, big viruses producing over 75 viral proteins. And again, many of the proteins in the pox viruses are used to modulate the host immune system. They have nothing to do with building new viruses directly. Indirectly, they contribute to that by helping to shut down the host immune system to allow the virus space to be able to replicate. So we've got a wide variety of viruses that are out there. I think just to kind of wrap up the talk and look at some key concepts that you can take away with you is that, again, when we're looking at viruses, we're talking about what's inside the virus. Is it DNA or RNA? What is the structure of the virus? Is it enveloped or non-enveloped? These are some stratification terms that we can apply to kind of help us understand viruses as we look at them, comparing a DNA to an RNA virus, for example. But whether it's DNA or RNA, whether it is an enveloped or non-enveloped virus, all viruses are going to follow a basic life cycle, which is going to be they must attach to a host cell that is permissive to allow the virus to get inside. Once they attach, they must be able to penetrate and get across that plasma membrane to gain access to the internal space of the cell. And once the virus is inside of the host cell, it has to have a way to be able to either escape from a vesicle that was maybe used to bring the virus in when it was internalized, or it's got to find a way to be able to get the viral structure that surrounds the viral genome destroyed, so the host cell replication machinery can get access to the viral genome. So it must uncoat. So we have attachment, penetration, and uncoating. And then once we have these three phases occur, we're going to have gene expression, which is going to lead to then the production of new viruses, which can then go out and infect new cells. And this concludes short course session number 2.